Thank you everyone for staying here. So today I'm gonna talk about our approach that we use uh, um, uh, to trying to bridge the gap between linear aligners and graph aligners in the area of whole genome sequencing when we are trying to solve the problem with building a population reference. So very quick background. So in whole genome sequencing, the basic task that almost all pipelines that need to do is to find a population reference or just a reference to do the alignment over. But this task by itself is not that straightforward because we know that the standard reference only represent just one haplotype of the genome, but but humans, for example, are very diverse. So we can have many variants in the genome that are not present in the reference. So in particular, we can think about some genes that are highly polymorphic. For example, the HLA genes, they are related to the immune system in the human body. So it's very important, but they are highly polymorphic. That means the set of genes can have a lot of alleles, and these alleles can be sometimes dense in variants, like this class 2 gene. That means that if any read that were sequenced from this allele, it will have a lot of mismatches that if we try to align it to the reference only. And this can cause a problem depending on the underlying, the downstream analysis that is being the target of the analysis. For example, if we are trying to do genotyping or HLA typing, this would cause a problem that we will miss a significant amount of the data. So luckily there are several efforts to trying to build a more comprehensive database for some known genomic variants. For example, the immunopolymorphism database that focus only on the HLA genes. And this database in particular have gone through a lot of growing on recent years that contain a lot of data, a lot of allele sequences that were found in individuals. So the question now is how we can, so with this amount of data, how standard pipelines can use this information to empower their results and improve their accuracy. So the standard approaches we can see now is either two directions. One of them is the linear approach, is basically the most, so it's straightforward. So along with the reference, we can just linearly concatenate or combine the alleles from the database, so we can align to the entire set using alt-aware aligners like BWMN. So this approach is, as I said, straightforward, and it uses all the current literature that that is present in alignment. But the main problem is that there are a significant amount of sequence duplications. Like all these areas, they are presented many times across the sequences. This can cause a problem in ambiguity that we'll have a lot of multi-mapped reads that can add complexity to the task that we are doing. The other approach that have been starting to gain more interest and a lot of effort now are trying to do is to do graph approach, and is to build graph aligners, where reads are directly aligned to a graph that were built based on combining this data. So instead of representing them as a linear combination of sequences, they are all collected into a graph, and the alignment for the reads are directly done over the graph. One example of that is the Hitseq genotype. It's a graph aligner. So the advantage is obvious that it saves a lot of this duplication. It's not needed. It's not present anymore. It preserves the structure relationship between the alleles that can be benefit in understanding what is going on in some analysis. But the problem that we can see so far is that these approaches are not mature anymore yet, and usually require a lot of time and memory. So this is where we are trying to, so okay, we have the two approaches, how we can bridge these two approaches together. So the simple question that we are trying to answer is for people who are working on this area, but they are doing the linear way, is it really important, or how desperate they have to be to switch to graph aligners? Do we really need to build graph aligners or not? So in other ways, is it possible to combine the advantage of both approaches? So we get the advantage from the graph aligners, but we still continue working with our linear aligners and avoid the time and space complexities. So our approach to bridge this gap is basically we are trying to segment, to do segmentation for the population graph. So simply our outline for the method, 3 steps. First, we build the population genome graph, and then we linearize it using into a set of segments, and then use this set of segments as the reference for the alignment step. And we'll go over each step very briefly. So, so the first, we begin from that we can get from the database is the allele itself. For example, for each gene, we have the set of alleles that represent that are provided in this database, and either they are provided. So, for example, HLA, the HLA database, they provide already the multiple sequence alignment, or if it's not present, it also can be derived using several tools that already exist. But for now, we began from the multiple sequence alignment. So this is how it looks like compared to the sequence itself. And then we build the multiple sequence alignment graph that represent this alignment and between the alleles. And we represent it as partial order graphs. This is the standard way, or it's commonly used to represent multiple sequence alignment sequences. And then we go from the graph into flattening it. This is a step that we need for our next step. So it's basically preparing for the next step of linearization. So it's basically as if like you took this graph, which consists of layers, and stretch it. So now we can have all the nodes on one axis, and edges can jump between nodes depending on the structure of the graph, instead of just edges between every layer and the preceding and or the next one. So the next step is so now we have this flattened multiple sequence alignment graph. So the core concept that we introduce is we can linearize this graph first before using it later. So how we can linearize it is basically we transform it into a set of sequences, a set of segments, and we adapt our approach that we introduced called Yanagi. This actually was built for transcriptome segmentation. It's using the same concept, but was for transcriptome segmentation, and now we want to use it for this problem as well, because there are common similarities between the two problems. Yanagi, in very brief, I will not go over the details of how it works, but very briefly, it processes a graph and it's segmented into L disjoint and maximal segments. And I will go over this tutorial very quickly to see for this example. So if we have 3 alleles, 2 of them have like indel and SNP. So if this is the flattened MSA graph that we achieved from the previous step, how the segments would look like for this example. So the first set of segments were created for the first allele will look like that, representing the 2 regions and this connection. While for the second allele, we will have these 3 segments that go over the graph itself, and the last segment will be added that come from this connection that, this one that came from allele number 3. So what I mean by the segments are L disjoint, that you can see there is no 2 segments that have overlap in the sequence itself more than L. And L is the parameter that our algorithm takes. It's usually correspond to the read length. So for example, if at the end we are interested in aligning reads of length 100 or 150 or whatever, this will be the value for L. And by maximal, we extend the segment as much as we can until we reach a branching point in the graph. So for further details of how this works, you can either see me in the poster or check the Yanagi work online. All right, so the final step, now we generated the segments for a set of genes that we are interested in capturing the alleles for it, which, for example, the HLA. So if this is the whole genome that we have, and we have a set of alleles for set of genes, so the final reference that we can use that were generated from our approach would be the entire reference as it is, but we replace the parts that correspond to the gene from the reference with the set of segments that were created from our approach. So this look like it's similar to the linear approach, but these sequences, instead of representing the entire alleles, they represent the segments that were generated from the graph. So very briefly, the experiments that we have done, the first question that we wanted to answer is how the segments would look like. The very first concern would be, since we are linearizing graphs, this can add a concern of the number of sequences that we will process during the alignment step. And we can see it's actually a linear growth, so it's non-exponential, so that's good. And the length of the segments so like 75% of the length of the segments generated, they are between L and 2L in the length of sequences. And this also can give us an idea that most of these segments that are short or less than 2L, they correspond to regions where variants are very close to each other, less than 2L. So that's where the graph will be complex, and the segments will appear to be beneficial. The last thing I wanted to show here is that's another thing that we were interested in seeing, that since some aligners use this K-mer-based approach to make it to make the alignment faster, or for indexing, or and so on. So we wanted to see just for the HLA genes, how much of the k-mers are actually new that were not present in the reference before, and we can see that it's around 30, and in average 35, 36%, which is significant if we think about this is amount of k-mers that were not present in the reference. So it can give us an idea how significant adding the alleles and segments for it. So very quickly, for the experiments that we have, that I'm showing today, is we focus on HLA read extraction. So basically the task is we want to align and we want to get the reads that were aligned to the HLA genes. How much of the reads that were actually coming from that were generated from HLA that we can extract using our approach. So we have simulated data for that with both reads coming from HLA and from the rest of the genome. So so we compare it with a graph aligner. So so we use recall for the metric. That's what we focus on for this experiment, is that how much of their HLA reads we can actually retrieve. We can see that with the graph aligner, it does really great job. It's almost retrieve all of the data. With BWMM, with only the reference, it's lacking behind. But when it's aided with the segments, it can go up back as with the graph approach. The other interesting part that is from our experiment is that we wanted to see how about we use pseudo aligners for that specific task. If we care about read extraction, we don't really need the exact mapping, location, and so on. And also with the segments, when we broke the gene into a set of local locations, so we can actually, if we do the alignment over these segments, this can give us the local information that people can be interested in, but still pseudo aligners, as you may have known, is that it's very fast, very lightweight. But before, using the reference only, it's really doing not really a good job, even compared to BWMM. But with the segments, it can go up again to the top. So that was a very interesting outcome that we saw. So at the end, to show the running time difference that between our approach and graph, and so using graphs to run for this for the sample, we have it takes like around 20 hours. While when we use segments, either with BWMM or even with RapMap, which is a pseudo aligner, it can do 10-fold speed up. So in summary, today we introduced an approach to linearize a population graph using a Yanagi segmentation, and it showed that the linear aligners can gain the power that graph aligners can do when they are aided with segments, while avoiding the expensive computation complexity from aligning over graphs. And this can show us that this actually opens the door for several applications. For if you have a problem where you have either the two approaches, the linear approach or a graph approach, to represent or to process multiple sequences. So this can be a good, using Yanagi's segmentation can be a good approach to bridge between both approaches. So for future extensions for our work, of course, we need to do more extensive analysis on different genes, maybe using instead of starting from the multiple sequence alignment from the VCF files directly. And two questions that we haven't targeted or answered yet: how we can handle complex repeats and complex structure variants. So at the end, I want to thank the team we were working on that project from University of Maryland and from Illumina, and thank all the audience for your patience and for your attention. See me at poster today if you have any questions, and we can talk. Thank you.
So nice talk. We went a little bit over, so we have time for 0 or 1 questions. But Mohamed is here. He's the last speaker, so you can come up and you have questions. You can ask him specifically. We're back here at 4:40, and I guess we have the keynote following exactly that. So please try to be back on time. Looks like 0 is the number that we're going to use.
All right, thank you.
Okay, thank you very much for your talk.